Today, we're going to be focusing on the cell membrane and looking at what it does and exactly what its structure is, what it's made up of. Okay, so the function of the cell membrane, there's three of them. Number one, it serves as the boundary of the cell and separates it from its aqueous environment. So aqueous means water-based, and no matter what living thing you're dealing with, the cell is going to be in an aqueous environment. Whether it's not like, that doesn't mean it's like living in an ocean or a pond. I mean, it might be, but like our bodies are aqueous environments. Plant structures are aqueous environments. Okay. Number two is going to help in transport materials into and out of the cell. And number three is going to be involved with cell to cell communication. So we're going to start by looking at this first one here. And so in order to understand how the cell membrane separates the cell from the environment, you need to know what it's made of. Okay. And so does anybody know what kind of macromolecule makes up a cell membrane? Maybe you remember back from your bio class, a lipid. Okay, a lipid makes up the cell membrane, and a special kind of lipid called a phospholipid. Okay, so a phospholipid is made of two primary parts. We refer to them as like the head and the tails, but obviously it's not like an actual head and not actual tails, okay? But the head portion here, now remember, we're dealing with a lipid. And a lipid is like kind of the funky one, right? It doesn't have like a specific monomer or polymer, but remember a lipid has a glycerol with some fatty acids, okay? And so here is our glycerol, okay, in the head. So the head has got this glycerol with a phosphate attached to it, okay? And then the tails have two fatty acids here, one that's saturated and one that's unsaturated. Okay, so phospholipid because phosphate lipid, right? Okay, so that's the general structure. And important things to know about this is head, just based off of the chemistry going on, is hydrophilic, water liking. It's going to be attracted to water. And the tails, however, because it's made of a lipid, we learned lipids are hydrophobic. So the tails are going to be hydrophobic. The head, because of this phosphate group, is actually hydrophilic. So one part of the molecule likes being near water and the other part of the molecule doesn't. And so the vocabulary for this is called amphipathic. Amphipathic, when you have a hydrophilic part and a hydrophobic part. So phospholipids are amphipathic. So let's think about now, if we have a bunch of phospholipids and they are in an aqueous environment, which setup here is going to make them all happy? Which ones are going to get the hydrophobic places where they want to be and the hydrophilic places where they want to be? What do we think? Our answer here is going to be this third one, okay? Because the heads want to be near the water. Here, right, we've got heads near the water, but guess what? The inside of the cell is also water-based. So here our heads would be happy, but our tails wouldn't, because our tails would be exposed to water on the inside. Here, our heads would be happy because they're on the inside of the cell where there's water, but the tails wouldn't be happy because they're facing the outside of the cell, which is water. But here we have our heads facing the outside, which is water, they're happy. Our heads facing the inside, which is happy, and the tails are in the middle, so there wouldn't be water there. There'd be this like water-free zone. Okay, and so this is why phospholipids form a bilayer, bi meaning two, okay, because it allows the hydrophilic heads to be facing water and the hydrophobic tails to be pointed away from the water. So both are like happy. Of course, they don't actually have emotions, but you know, you can think of it that way, okay? So we've got this bilayer, okay, we've got two layers of stuff, okay, and there's not just phospholipids here. There's lots of other stuff going on, okay? Look at all these. Oh, we got some proteins. We got some cholesterol. We're going to have some carbohydrates. We're going to have all kinds of stuff that's also embedded or connected to this bilayer, okay? Back in 1972, these two scientists discovered what we call the fluid mosaic model. And what this says is that the bilayer is fluid, meaning it's moving. So individual phospholipids and proteins can move their position. They're not just stuck in place. Some things are kind of anchored, but other things move around. Okay, it's a mosaic because kind of like a tile where we can see lots of different areas. As we saw in the last picture, there's lots of different things going on in the phospholipid layer. So we got lots of kind of patterns or things happening. But like, how did they figure this out? How do they know that stuff moves? Well, it's pretty crazy. basically what they did is they like took some fluorescent dye and colored some surface proteins on the bilayer of mice a certain color, and they did the same thing with a human cell, and then they fused those cells together, and like they waited an hour, and then they looked at the cell membrane, and all of the colors were all mixed together, showing that those like proteins moved around and moved through the phospholipid bilayer, so they're not necessarily all like just stuck in place. Kind of cool, huh? So the membrane is fluid, it's moving around, but here's the thing, some things move around, but other things are like connected to like the extracellular matrix, and so they're going to stay put. Um, remember back with lipids, how we learned that unsaturated fats spread things out, so they're like oftentimes liquid at room temperature. Well, here are those tails with the bends in them basically help keep that membrane fluid because the membranes won't be able to pack as closely together. Those phospholipids will be spread out naturally. Okay. And also temperature. Hopefully, you know that like when things get cold, they're going to move closer together. And when things get warm, they're going to spread apart. And as you can imagine with the cell membrane, if it were to freeze, that'd probably be bad. And if it were to get too hot, things could spread apart too much and like the cell could kind of lose its boundary. So what's our natural defense for that? And the answer is actually sometimes cholesterol, which is kind of interesting. Cholesterol has this awesome ability of it keeps cell membranes fluid when they get cold, so it keeps things spread out, but it also helps hold things together when it gets warm, which is kind of awesome. And there are certain animals that will have like slightly different compositions depending on their environment. So like fish that live in super, super cold water, like freezing cold water, they have way more unsaturated fatty acids to help them prevent from freezing. And then like things that like live in like thermal springs have special lipids that keep things from getting too fluid and disintegrating, which is neat. Okay. So this structure of these lipids and these proteins is going to allow some substances to cross the membrane. Some will go easy, some will need some help. And next lecture, we're going to deal into more of which things can pass and what kind of help they may need. Okay. But let's take a look. Back we saw at that picture, right? We saw that there's lots of different things in the cell membrane. Let's take a closer look at some of these proteins that could be in the membrane. Okay. So we've got two different kinds of membrane proteins. One is an integral protein. So like this right here would be integral, this would be integral, and right now they're colored in red. So they go either all the way through both layers of the cell membrane, or they at least extend into the interior where the tails are. Okay. Then the other type of protein is called a peripheral protein, and this means that they're going to be bonded onto the phospholipid heads. So they're not going to extend into the interior. Right? So why are proteins so important, and why are proteins the perfect macromolecule to have in the cell membrane? Remember when you looked up the R groups and you saw that some were polar and some were non-polar, and some were hydrophobic and some were hydrophilic? So proteins are going to be made up of lots of amino acids, and they're going to have different R groups. And so guess what? The parts of the protein that like touch the water side, either the inside of the cell or the outside of the cell, right here they're shown in purple, those are going to be polar and they're going to be hydrophilic, water liking. The parts of the protein that are in the inner area where the tails are, they're going to be nonpolar and hydrophobic. So they're going to fit right in with those tails. So because they're amphipathic, they can not only fit in the membrane, but they can extend to the outside or the inside of the cell, and all parts of the protein are then happy, which is pretty cool. Like we've mentioned before, proteins do all the things. Here are some of the things that they do as a reminder. And one of our membrane functions is cell-to-cell communication. So we're going to specifically look at this right now and see how that works. So here's an example of a membrane. And you've got these carbohydrate chains. You can tell that because they're rings, okay? So you got these carbohydrate chains coming off of a protein. These would be called glycoproteins, glyco for the carb, okay? So we've got these glycoproteins. And what they do is they're actually really important with cells identifying themselves. So, like, your immune system is constantly going around and basically like touching the surface of the cells, and it recognizes the patterns, it recognizes the shapes of the glycoproteins. And so it'll touch cells and go, oh, yep, that's me, that's my body. Oh, yep, that's me, that's my body. And then if it comes to a glycoprotein, it doesn't recognize. It goes, oh, we have an intruder. Okay, come on. And it draws other immune system cells and they identify that intruder and they destroy it. Okay. So um, so my glycoproteins would be different from your glycoproteins. Not only that, my heart glycoproteins would be different from my liver glycoproteins. Okay. So think about an organ transplant. So if I need a new kidney, how's that going to work? Wouldn't my immune system then go and feel those glycoproteins and go, up, intruder? And that's exactly what happens. So if people get an organ transplant, you might have heard of organ rejection. That's where the person's immune system identifies that new organ as an intruder because the glycoproteins don't match. And so their immune system tries to attack it. So they're on some immunosuppressive drugs, some anti-rejection drugs that basically tamp down your immune system so your immune system doesn't start attacking that brand new organ you just got. Kind of crazy. Last but not least, I do want to mention the cell wall because some things have a cell wall, right? Not animal cells, but plants do, prokaryotes do, fungi do. And right, the cell wall, if you remember, surrounds the cell membrane. Okay, so the cell wall is not made of a phospholipid bilayer. It's made mostly of cellulose, OSE. That's going to be a carbohydrate. But also it's going to help plants even more so with determining that boundary. It's going to help give plants and these other things that don't have bones some structure and shape. And your cell walls are also semi-permeable as well. Okay, so as always, here are the things to consider. Here's what you should know about this topic, and here's where we are in the textbook.